Welcome to the Future of the Planet series. And this series is about the sustainability of the planet and the technologies and the businesses of the companies that are making that possible, brought to you by IndieBio. And so the session today is about drug manufacturing. And we've got three very special guests joining us, which I would like to introduce now. First up, we have Dr. Jeffrey Ling, an MD with a specialty in neurology, also a PhD, a retired colonel of the US Army who served in Afghanistan and Iraq with a huge number of awards, but I'll just mention a few right now, from an award, the Department of Defense Medal for Distinguished Public Service, the Legion of Merit, the Order of Military Medical Merit. Since retirement, things get more busy because Jeff joined DARPA, became the founding director of the Biological Technologies Office, and he is now CEO of On Demand Pharmaceuticals. It is truly an honor to have you join us, Jeff. And next up, we have Sari Goodlevaletti, who's the global head of R&D for a company called Dr. Reddy's Laboratories. And this is calling in from Hyderabad, India, 9:30 their time, which we're grateful for you to join us, Sari. So for people who have not heard of Dr. Reddy's Laboratories, that is a global generic pharmaceutical company, the ninth largest in the world, publicly traded on the New York Stock Exchange, as well as the two exchanges in India. And generic medicines are the ones which have expired, where the patents have expired. And in the US, over 90% of drug utilization, drugs that are prescribed, are generic medicines. And they're important for access and patient access because of affordability, because they comprise less than 10% or a little bit more of drug spend. So that's very important. Lots of such medicines are manufactured, and it is people like Sari and his colleagues, as well as other companies, that make that possible. Welcome, Sari.
Morning.
And finally, we have Aidan Tinafar joining us from Toronto, Canada, CEO of Librum Biotech, which is a company now in the IndieBio Accelerator program. Aidan has got a very interesting career path and actually gave that up because of his passion for synthetic biology, which eventually led to Librum, which we will eventually hear from. Welcome, Aidan.
Hi, thank you for having me.
So here's how this will work. We have our audience out there. Please type in your questions on the chat box, and myself or my host assisting in the background, Alex, will be able to present these questions to me, and I will send them to our panelists. And I will get started with a question to each of the three. And I will start by saying that 2016 was a pivotal moment for all three of you. And the reason for that, I think, is because that was when you started your journey on a particular vision that you had about the future of drug manufacturing. So I will start with Sari with my question to you, which is fairly long until you can turn it over to you for the general answer. So you started, if I got this right, you went to the IIT Madras, the Indian Institutes of Technology, which is a network of the premier engineering colleges in India, the one in Madras. Went on to the Massachusetts Institute of Technology for your master's in mechanical engineering. You joined corporate America and companies like GE and Qualcomm, became a management consultant thereafter at McKinsey & Company, and then joined Dr. Reddy's Laboratories. And in 2016, you became the vice president of R&D. And in a company where innovation is important now, as I understand it, titles are no longer vice president. It's more a description of what you are really doing. And your title now is global head of R&D, leading over 1,000 research scientists who are developing hundreds of products at any one time, which need to be scaled up in a way that allows hundreds of millions of doses, altogether perhaps billions of doses of each of those products all combined a year. And you, in your description, say that you are transforming pharma with data, digital, and analytics. So Tell us about your vision. How are you making this more possible for pharma manufacturing in the future and making quality products better at such a huge scale?
Sam, thank you very much for the introduction and for the question, and it's great to be here. See, pharmaceuticals is a sector which relies on very large volume production of individual unit doses, and it needs to adhere to very high quality standards. In particular, generic pharma is an industry which is an order of magnitude larger in terms of scale. And that's because we aim to produce the same drugs that innovators produce at a fraction of the costs, in many cases less than 1% of the cost and the price as well. So just as an example of scale, we currently produce 5 billion tablets a year, tablets and capsules and oral dosage forms a year, probably 10 million vials or units of injection a year, and across maybe 120 different individual drug products just for the US market alone. And the US is half of our business. So there is a huge role for data to play in this. One, during development, we currently work on about 200 individual products at any given time. So it's very important to have huge amounts of data rapidly acquired and processed, so we know that the drug substances and drug products that we're making, we understand the chemistry, we understand the formulation, we understand the biology completely well, so that we can make a high quality process that can be transferred to manufacturing. In manufacturing, it's very important that we're able to produce the drug products to not just 6 sigma, but even 9 sigma quality levels. That's because when you're producing 5 billion tablets, you need to produce them at a very high accuracy and quality. What I found interesting in this industry is there's a huge amount of testing that goes on, right? Every batch that you produce, and a batch could be as much as 1 million or 2 or 3 million tablets. Every batch that you produce is sampled and tested. And based on that, you statistically decide that the product is meeting all the quality requirements in terms of purity, in terms of the potency of the drug, in terms of how it's going to release inside the human body. There is a role to play for digital and data to understand the relationship between the manufacturing parameters. Essentially, it's something similar to multiple other industries, either the automotive industry or the semiconductor industry, which have mastered the art of producing millions of individual units at very repeatable quality levels. And I believe that those technologies and those understanding of data can be brought into the pharmaceutical industry so that we can also produce billions of dose forms at very high quality. That should eliminate the need for repeated testing, right? Or should reduce the need for testing and give higher assurance of quality. So the journey that we are undergoing is, one, a very deep understanding of how the input materials, the input processes from R&D and in manufacturing affect the quality of the drug. And once we are producing large quantities of drug, how do we give high degree of assurance that the products are meeting the quality requirements, even beyond what we can test them?
Thank you. My question next is to Jeff, and you, what I see here is that you joined DARPA and was founding director of the biotechnology, Biological Technologies Office, one of the 7 technical offices in DARPA. And there was a huge list of different projects that were conducted there, but there's one particular one which was is relevant to us, which is the one on on-demand pharmaceuticals. You put out some applications for which people could apply to. MIT, I believe, was one, but there were others. And that became a hugely successful program, and that's led to further work now. Can you tell us about what led to that idea, and where is that going?
Yeah, no, absolutely, Sam. Thank you for this. And it's wonderful to be on a panel with Sari and Aiden, the 2 very eminent folks. And so I really enjoyed that, Sari. Thank you. So it all started back in 2003. So in 2003, I was sent to Afghanistan, my first tours of duty overseas. And I was sent as a doctor. I'm actually an ICU doctor, intensive care unit doctor, and I was running the ICU in Afghanistan. And a young service member came to me, and he was pretty badly hurt. And he suffered from a number of things, but most relevant for this discussion, he suffered something called dysautonomia. And dysautonomia is where your blood pressure and your heart rate are just out of control. And so I could not put him on so we were in a tent hospital basically, and I could not put him on an airplane to send him back to Germany because it's a 21-hour flight with his heart rate and blood pressure totally out of control. That just you wouldn't survive it. And I needed a drug called bromocriptine. It's very easy to treat dysautonomia if you had a drug called bromocriptine. Bromocriptine the patent was issued in 1968 by Sandoz, and it's it's a generic drug like Sari was talking about, and I could probably buy it from Sari for about a nickel a pill. But at the time in Afghanistan, I couldn't. It just didn't exist. It just wasn't there. So I had to radio back to Germany, and the folks in Germany put a box of bromocriptine in a pocket of a jet pilot, and they flew it to me. So when you think about the cost of that particular box of bromocriptine, just in gasoline alone, it was, it's crazy. And so I was sitting there thinking to myself, geez, Louise, I mean, I have a PhD in pharmacology from Cornell, so I sort of know a little bit about what I'm doing. And I said, if I had a chemistry set, I could make my own bromocriptine. I mean, after all, it's an organic compound. It's made of carbon, hydrogen, and oxygen. It's got a little bit of nitrogen in it, I get it, but basically it's made of carbon, hydrogen, and oxygen. And if I had a chemistry set, I could make it, right? And in fact, if it's only made of carbon, hydrogen, and oxygen, and a little bit of nitrogen, if I had a pencil and a glass of water and an egg, I should be able to make it, right? That's all I need for starting materials. A little more difficult on that, right, Sari and Aiden, but you get the point. So the idea was, can I come up with an automatic chemistry set that would make me these drugs, particularly since the problem is bounded. I don't need that many starting materials. I made a carbon, hydrogen, oxygen, a little bit of sulfur, maybe a little bit of fluorine, maybe a little bit of nitrogen, but by and large, I don't need the whole periodic table. So when I came home from the war, I was lucky. I came home from the war and they said, well, you get to join this place called DARPA and you get to run with some of your crazier ideas. And so one of the ideas I had was, can we actually build one of these machines? And so if you fast forward, we succeeded. And this, I will tell you right now, is a military asset. It's not, it is purely 100% United States Department of Defense. The idea is to have a machine that can go on board a ship, go on board a hospital like I was at, called a combat support hospital, go out with a special forces team in some village and make medicines that they would need. And with some very simple starting materials, this automatic chemistry set will make whatever medicine they need. Now, I'm not talking about APIs. I'm an MD. I have never prescribed an API in my life. I've only prescribed a drug. So this machine has to go from starting material to a final product. And so the pod machine, what we nicknamed it, the pharmacy on demand machine, actually starts with some common precursors. It will make starting material. It will then go to make an API, and then it'll turn it into, in this particular case, a tablet. And the machine is there for the United States military, and it uses continuous manufacturing processes. So because they are meant for U.S. service members, we want it to be of the highest quality. So we measure the spectrographic fingerprint of every single pill that comes off. Every single pill. Every pill. Let me say that one more time. Every pill. So that way my service members are guaranteed to have the highest quality medicine. Even better, because it's for the U.S. military, we cannot be dependent upon foreign friends, such as India, or maybe foreign potential adversaries, which we will not name, for our starting materials. So our machine actually begins with domestically sourced starting materials that we get from the domestic petroleum and the domestic agriculture companies, because our U.S. military simply has to make everything from scratch, basically, using U.S. domestic source material, because we are the U.S. military. We cannot be dependent on anybody else for anything. I'm a soldier. You may not like to hear what I had to say, but tough.
Thank you, Jeff. And Aiden, so interesting story. In 2016, there was some publications that came out in some very prestigious journals. And these publications came from two academics, Jim Collins, not the business Jim Collins at Stanford, but the Jim Collins who is at MIT and the Wyss Institute at Harvard, who is a pioneer in the area of synthetic biology. And there was a postdoc at the time called Keith Pardee working with him, and Keith was the lead author in the paper. And they made a paper-based test for Zika virus, and it was written all over in the Atlantic, for example, as well as other well-circulated public publications. And also in Cell, the prestigious journal Cell, about something called cell-free protein synthesis, where they demonstrated that with antimicrobial peptides and vaccines. So what does this have to do with you? Well, I see that you did your business degree, and then you did a law degree, and then you articled and became a corporate lawyer. And then you decided that you wanted to go, as I understand it, back to learn about synthetic biology, and went back to school and did an undergraduate science degree in synthetic biology. And then in 2017, 1 year after all that news came out, Keith Pardee had now become an assistant professor at the Department of Pharmacy at the University of Toronto, and you joined his group. And from there, fast forward to today, you are the CEO of Liberum Biotech. And I see the tagline is "Making proteins have never been easier." So tell us about what got you into this. What was so appealing to change the trajectory of your life? And how did this company come about? And where do you see your vision going?
Yeah, thank you, Sam, for that well-researched question. And thanks to the panelists for their incredible stories. That was amazing to hear. One day I was going about my day as a corporate lawyer, and I asked myself what I would do if I won the lottery. And without ever having won anything, I went ahead and did just that. So I went back to school to study molecular biology. Then I heard Keith Pardee give this incredible talk about the power of freeze-dried cell-free gene networks, and that's part of the Zika paper and also the 2016 cell paper that Sam mentioned. And it was true magic with pixie dust and everything. And so, you know, back in the late 70s, there were probably a handful of people in every town who could program computers. Now every child can learn to code. And the same shift is happening with engineering biology. I'm certainly an early benefactor of this shift, and we've seen a glimpse of this new wave of biological programming with the iGEM competition. And so since the 80s, drug discovery has become increasingly more expensive, with the cost of developing a new drug doubling every 9 years. And so, and we've also seen with the current pandemic that there are bottlenecks in drug discovery and scale up. As we learn more about diseases, we're also realizing that this one-size-fits-all approach is not very effective. So there's a large group of people, including myself, that believe synthetic biology is the thing that can get us through these problems by helping lower the barriers to drug discovery and also offering ways of making old drugs. Neri Oxman, who's a professor at the Media Lab, tells this fantastic story about a fruit-bearing tree in heaven where the whole of the tree is the fruit. And to me, that's a perfect analogy for synthetic biology's central thesis: efficiency, low waste, and a reminder of the real power that biology has. So after going back to school, I received a small grant to express and purify 2 therapeutic proteins. And remember, this is pre-Liberum days, so I needed lots of equipment to do that. And I asked Keith if I could use his lab to make the proteins. And so he did me one better. He hired me. And then he asked me to join his lab as a grad student. So over the past 3 years, myself and a team of incredible scientists at his lab have worked tirelessly on bringing this magic of push-button protein expression to research labs around the world. And as you know, discovering even a single new therapeutic protein can launch billion-dollar industries. So we want to allow researchers to do that consistently. Steve Jobs has this quote where he calls a desktop computer a bicycle for the mind, and because they both let you get where you want to go faster with much less energy. And now we can do that with proteins. In 30 years, when we look back at the synthetic biology revolution, we want to be one of the things that accelerated that process.
Thank you, Aidan. So I will, we're opening up the questions. You've heard a little bit about each company and each person here. I will start with, let's see, I hear there's a question here, and let me provide the background and framework to make sure this comes out properly. Continuous manufacturing. So continuous manufacturing, I think, was pioneered, maybe not, by Henry Ford. Imagine an assembly line where cars are assembled as it's moving along, so it's basically continuous. And almost all industries do that now for manufacturing of products, except the pharmaceutical industry. It is still made in batches. Imagine batch making recipes in your kitchen. That's how it is. You put it into in your kitchen. It's a mixing bowl, but pretty similar in a pharmaceutical company in a reactor. And then you move it to something else, and then something else, and then you make tablets. But there has been a move, also supported by FDA, as well as other regulatory agencies around the world, to get into continuous manufacturing, which is not easy because we have good manufacturing processes and there's a method in place for that, and you got to comply to that. But companies are moving that way. So I will actually ask, I'll ask Sari first and then ask Jeff, because I suspect both of you have some aspect of that. And I'll start by Sari, and then Aiden, if you actually do something like that yourself, we'll ask you too. So Sari, tell us about that. What's going on? Educate us more, because not everyone knows all about this.
Sure. Thanks for that, Simon. It's a very exciting field. And Jeff talked about that, how on site in a war zone, you can actually start producing drugs in a small footprint. And that's what continuous manufacturing is all about. It's about shrinking the footprint in which you produce a drug or any chemical for that matter. And many industries already do that. So what continuous manufacturing is? Typically, if there are 3 or 4 chemical reactions, let's say there are 4 chemical reactions and purifications have to happen. What happens, the larger the quantity that the larger the reactors is. So you will end up having 5,000 liter or 10,000 liter large reactors, and you'll have one for each process step of the reaction and the drug making scheme, with very large equipment which can do the filtrations and dryings and crystallizations. This is good when you're producing large quantities, but typically for one of our big drugs that we produce, we make about 180 tons of the active ingredient per year. This is a drug called atorvastatin, which is a very common drug in the US. Lipitor is the name of the drug. We make 180 tons of the active ingredient per year. Now imagine that's basically a few large buildings across 3 manufacturing plants that we need to do this. However, what you can do is you can make the same drug in what is called continuous flow. You introduce all the reactants, A plus B gives you C, then you introduce a D, all the reactions in a pipe, and you essentially have the reactants flowing at a very, very high speed through the pipe. And as they go through the pipe, at every stage, you introduce the additional next reactant and the next reactant and the next reactant. So what happens is you are not, at any given point of time, you are not holding the entire 5,000 liters of reactant in a stationary large vessel, but it's moving very fast. And as it moves fast, it's converting. And so if you want the same quantity, you can do it in maybe 1/10 of the footprint by just running it longer. So you end up getting a continuous output of either a liquid or a solid chemical at the end of the process. Now, this is very exciting because it allows you to shrink the footprint. It allows you to turn production on and off. It allows you to make multiple drugs in a smaller footprint because you run it for a week, you produce one drug, you stop it, you clean it up, you produce another drug in just the same week, and so on and so forth. This is something that many companies, the innovator side and generic side, are investing. This has the ability to reduce the cost. This has the ability to reduce energy conversion. It has the ability to scientifically control reactions, which in a large vessel you cannot control. Imagine a reaction which is either exothermic or endothermic. The amount of heat load that you're going to put on your reactor is very, very large. Whereas if you do it at a very small scale and rapidly moving reactant, you shrink all of that very quickly and you can control the reaction. It's kind of like a nuclear bomb that you can just control without really going off. Right? So when you can do all of this, you achieve greater degrees of cost efficiencies, scale efficiencies, and also process control. Now, what is challenging about this is now you're essentially building a control system with a very high speed set of reactions taking place in sequence. And a perturbation in any one step can start showing up downstream, right? Either in terms of the quality of the raw material or a particular temperature, energy control, etc. So this becomes a very complicated system where you need to understand the kinetics of your process reactions, the speed with which the chemicals react, and what affects the speed with which the chemicals react. If you heat it up a little bit, if you cool it down a little bit, understanding this at the R&D stage becomes very important. And when you understand that and put it in the system and build a control system, which can essentially control all of these reactions simultaneously, you end up being able to take advantage of the benefits I talked to you about. So that's what continuous manufacturing is. It's very exciting for innovative pharma. It's very exciting for generic pharma. And maybe I'll hand over to Jeff, who's actually practiced this and taken advantage of doing this on site.
That, Sari, was beautiful. I have nothing to add to what Sari said from a to explain. He did a magnificent job. What it does, continuous manufacturing does, is actually the biggest benefit you get is being able to shrink the footprint. And by shrinking the footprint, you're able to pivot. If you're in the situations that I explained to you, you want a manufacturing platform that can do a couple of things. Number one is make a certain amount of product, not metric tons, but rather maybe thousands of pills rather than billions of pills. That's one. Number two is you'd like it to be flexible. You'd like to be able to say, gee, I'd like 50 pills of X and I like 70 pills of Y and 10 pills of W. And a small machine can do that. We've actually showed it. We can actually go from one product to another product in a couple of hours. And soon one day we'll be even faster than that. But right now, turning over a large factory, such as Sari was talking about, would be very difficult. It's not easy. It's not a trivial thing. Whereas at a small scale, it is actually fairly trivial. So it allows you to make multiple things on site. That's another thing that you have that's good. And number three is, most important, is the footprint size. So the machine that I spoke to you about that we're building for the military is about the size of a household refrigerator. That's all it is. It's about the size of a household refrigerator. You can see it, you can look at it, and it does not, let me be clear, it does not do what Sari's company does, which is make billions of tablets for the world, all right? My little machine probably will make 2,000 Cipro tablets in a day for a combat support hospital. But depending on the drug, you can actually get decent output. We can make 40 million doses of fentanyl, for example, in a day. We can make 2,000 doses of midazolam in a day. I can make 1 million doses of atropine a day. But I can only make 20,000 doses of propofol a day. So it really depends on the medicine and how you would use a platform like this, which tells you how continuous manufacturing may be in fact beneficial. If you're going to make 5 billion doses of Lipitor to meet the world's need, this is probably not the best way to do it. You know, Sari's way is probably much, much more efficient. So it is not a matter of either or, it's a matter of what is the application space. But the scientific explanation that Sari gave was wonderful. I should have tape recorded it. I should play it for the students and the new hires that I get so they can understand it. But that was again, Sari, that was a beautiful explanation.
Thank you, Jeff. And I'll actually also ask the same question to you, Aidan, because we actually have another question related to that, which is, can on-demand technology generate biologics or just chemical drugs? And I'll preface that a little bit because you're making proteins, I think. But back in the day, peptides, which is a smaller version of proteins, used to be even made non-chemically if it was large enough through a recombinant E. coli system, and you have to ferment it. Now you can make it chemically, almost, in some technologies. Now you, Aidan, are going to proteins. And so I imagine it's self-free, so you're not doing stepwise. Maybe it's some other biological version of continuous. Can you tell us about that?
Yeah, so I'm just, those were incredible explanations of the process, but I'm just going to add this slight thing that's also happening on the biological side of things too. So on the cell-free enzymatic reactions and also on the cell-based systems where there are a few IndieBio companies that are doing incredible work in terms of continuous manufacturing using cells, and they're getting significantly higher yields because the cells are in a better condition to produce, so that they can actually really reduce that footprint by 2 orders of magnitude, something like that. And so, and yes, at the same time, you could use these type of scalable systems to produce biologics as well. And so, unfortunately, with the peptide synthesis systems that are currently available, I think the best one right now does 160 amino acids, 140 to 160, with 99% accuracy, which means at 140th amino acid, only 20% of your product is actually made accurately. But if you're using enzymatic systems, these have been developed through evolution, through billions of years. So the accuracy and the fidelity to what you're actually building is quite high. And so that's the type of system we're bringing into our technology. And so, the cost of DNA synthesis rapidly fallen. So basically you can order any sequence that you want online very cheaply, put it in this type of system, and it'll give you a purified protein within a few hours. And then if you compare that with the traditional systems of having to grow large batches of cells, fermented, all the equipment you need, all the expertise you need, all of that is going away soon. And these types of automated systems are definitely on the horizon.
Okay, we have another question that has to do with waste product. When you're doing something on a bench scale, but since we're also talking about continuous, maybe there's a savings of waste, I don't know. How can waste products be safely handled or disposed of in a batch or bench top drug manufacturing? So Jeff, maybe if you're in a combat field, I don't know what's coming out of there, but you got to get rid of this stuff, or maybe you don't because there isn't a lot. Tell us about that.
Yeah, so the nice thing about continuous, since you are working on small batches, it's actually very efficient. We actually produce 2/3 the less waste product than traditional batch processing because of the way that, as Sari described it, you're running through things very quickly, and you actually end up regenerating some of your reagents. You end up burning off a lot of your reagents because you run at very high temperatures and so on and so forth. So a lot of the waste is actually reduced if you actually take a look at these different processes when you use continuous processing. You are going to have some waste, there's no question about it. You still have to manage it. But the continuous process we have, and we've actually published this, is actually reduces waste by 2/3. So it's actually a fairly significant savings, in my opinion. It's a reasonable an approach, particularly if you are in an austere environment, such as the ones I'm talking about.
Now, on a large scale, I've seen there are companies out there like Vertex, I think was one of the first. Lilly is getting into this, J&J. Now they are pumping out hundreds of millions of tablets, I think. My understanding, if I may or may not be correct, is that you do need to stabilize the system in a continuous process when you first start it up. Like, it's just like when you run hot and cold water through your faucet, you let it run for a bit before the temperature stabilizes to a point where it's at the temperature you want. But in pharma, it will stabilize to a process. Is that very wasteful? I don't know if anyone has experienced that. Sorry, is what's going on there? And is that a lot of waste?
Sam, that's a good question. I would say overall, there will be a little bit of waste that occurs, right? Either during the initial startup or when there are perturbations, you might have to divert the reactant into a buffer tank so that you're holding something while you check out what's going on. But overall, if you see that over the large scale, if you're producing something, ideally these plants are intended to run for weeks on end without interruption, right? So if you imagine that the first 30 minutes, you're going to produce a product which is to be discarded, as opposed to something which is going to run for the next 7 days nonstop. It's a very, very small percentage. If you do it right at the right quality, this will be far less than the amount of material that has to be discarded in batch manufacturing if the batch goes wrong. The trouble with batch manufacturing is if you find something that went wrong in the batch, the entire batch of 1 million tablets or 10,000, 5,000 kilograms of the drug substance might have to get discarded. Whereas in continuous, what you do is, if you find that there's a perturbation or something non-standard, you discard the material produced for a few minutes, you divert it away and say, we just continue to go through. So I actually believe there will be less wastage and better process control.
Thank you. We've got an interesting question here, and I'm actually going to preface it with transportation, because Jeff, you mentioned about a fighter jet being able to be commissioned to actually deliver a single drug. I saw some interesting information about this pharmaceutical supply chain, quoting Julian Suchier of the Emirates Sky Cargo Pharmaceutical Division, that he says that if you want to transport 1 million doses of for instance, vaccines for COVID, you would need a Boeing 777 that can it will take a Boeing 777 airplane to deliver 1 million doses. For half the world population, you would need 8,000 cargo planes. And right now, Emirates alone is only running 70 cargo planes, delivering all kinds of other hardware as well. And so distribution is a very, very big challenge, but a lot of the global supply chain is predicated by having a single supply source where all this stuff has to be sent through distribution, through airplanes, through trucks, and it's all time limited. The question is, how challenging is it to have a single manufacturing system for drugs where you have regulations in different countries? Is it possible? Will perhaps the distributed system that you have, Jeff, make it possible, but then you'd have to scale it up? I'll start with Sari, because you're actually doing this for many different countries.
Yeah, so that's a very good question. The COVID pandemic actually has exposed some of the downsides of having highly centralized manufacturing, right? So the advantage of highly centralized large manufacturing plants, like the ones that we have in India and other countries, is that you bring huge cost efficiencies. You bring, essentially scale. Also, you don't end up spewing waste material all over the place, right? So you can contain where the waste material is going, and you can discard that safely. However, in the COVID pandemic, the day India went into lockdown, which was around the 23rd of March, we had to make a decision. Do we also take a 2-week shutdown, or do we keep going? And we said we will keep going, right? Because just because the pandemic hit doesn't mean people are going to stop taking other medication. So this has been a very difficult time for the pharmaceutical industry. And what that has done is various countries are now enacting laws that require you to have some manufacturing footprint locally, so that in the event of a global supply chain disruption, supply is assured. Having said that, that is going to make the cost of medicines go up, right? Because if I have to shrink my scale and have the same drug made in installments in 5 different plants, the cost will go up. So I believe the regulatory framework will evolve, but perhaps one of the upsides, the silver bullets, is in countries like India, which do continue to produce these drugs, have maintained their supply. There has been no disruption. And has gone up, but we and other companies in India have managed to supply 100% of the increased demand that has come in. So that may also give some comfort to countries that, look, at the end of the day, it's not going to all break down. But yes, there is a merit in some situations for local manufacturing as a backup, and that can be done.
Thank you. Jeff, I have a question for perhaps you, because this is more general about large and small scale systems, but I think Sari pretty well covered some general aspects of the large scale. How flexible are starting materials when you're actually using a small scale system? Can you actually get it from a different source? Is it actually a different material altogether? Are you getting it from some other source in the supply chain?
Yes. So the short answer to your question, Sam, which is a very interesting one, is the short answer is yes. The continuous process that we're using allows us to go earlier in the synthetic process. So you don't have to make each step of a traditional batch process is significant. I mean, you need large reactors and so on and so forth. At small scale, maybe another reactor, and the reactors themselves are so small, right? The continuous reactors that we use, for example. So the reactors we use, for example, are about the size of a, I guess almost like a book, a cover of the book, and about the thickness of just the cover. So they're actually very small, which is kind of nice, and allows you, therefore, to increase the number of steps that you want to use. And that allows you to go earlier and earlier into the synthetic process. And it allows you then, if you're earlier in the synthetic process, it allows you to do a couple of things. You can use really raw chemicals. So chemicals that don't have a shelf expiration date, right? And that's the goal in the military, is to start with things that don't have a shelf expiration date. Table salt has no shelf expiration date, for example, right? And so if we go earlier in the synthetic process, which this is all about, is that you're able to use raw chemicals that can actually be commodity chemicals too. So it actually helps you cut the price. Not as low as what Sari can get because he works at a very large scale, but it brings it to a more reasonable amount for our uses. What that means, Sam, is that with starting early in the synthetic process means that there are many common starting materials that you can use that can give rise to many different types of medications. So you're not hamstrung by having to start with a one-to-one, that you need this starting material this precursor to lead to this API that then leads to this final formed product. Instead, you start with this very early commodity raw chemical, and it can give rise to many different precursors that in turn then can give rise to many different APIs. So it actually simplifies the supply chain quite a bit. That's the whole goal of the military. And the idea is that you would have non-expiration starting materials. You would pre-position your machine to use it where you want. And it has all the benefits and the negatives that Sari had eloquently talked about. But from a military standpoint, you can understand it. This can go anywhere, anytime. It can go on board a ship. It can go into the jungle. It can go out into the desert. I mean, that's what the military wants. They want a capability that they can take with them. The other thing the military does do is the U.S. military doesn't go anywhere without the ability to make electricity and make water. So the U.S. military can make electricity anywhere, can make water. In fact, people don't realize that during the Haiti earthquake, the first assistance that the U.S. sent was actually a U.S. submarine. And you would say, why would you want a submarine to go to Haiti during an earthquake? You know, what are you going to torpedo? Well, the answer is nothing. But what does a U.S. submarine have? They have a nuclear reactor. And so that nuclear reactor was able to provide all the power that was needed to power the harbor and the airport of Haiti that allowed other people to come in. The second thing it allows it to do is they make 70,000 gallons of fresh water a day. 70,000 gallons of fresh water. And you're in an earthquake situation, you want fresh water. So the U.S. military, because it has its own electrical and its own water supply, it wants its own medicine supply to go along with it. So it actually fits into the concept of the operations of the military. And it's very much a distributed network. The other thing too is it's very resilient, isn't it? If you sadly got attacked and it damaged your machine, you would simply call to the next unit that may be over the hill and they have their own machine. And so this distributed network cannot be taken out, right? It's very, very resilient and it's very flexible, as we previously talked about. So from those standpoints, Sam, that's where starting with very common starting materials add to that story of flexibility and resilience, which is very important to the U.S. military.
Thank you, Jeff. Aidan, I have a question for you because you are, right now, you are at the cutting edge of where the technology is capable for cell-free synthesis of proteins, which is in the biological space much more complicated than small molecules. And right now, if I understand it properly, you initially will provide this more for research use, one would imagine, because that's where cutting edge stuff starts. But what's your vision for where this is going to go when it goes beyond the research laboratory? Of course, there was the talk about Zika virus, but that was more of a diagnostic. But the lab that you came from is working on all kinds of these things, which they call on-demand production of pharmaceuticals, but in the biologic space. What's your vision of that, and how might your machine be used as a use case there?
Yeah, so just as a slight addition to that question, it's just because proteins are made doesn't mean small molecules are out of the question using biological systems. So in that 2016 paper, they actually demonstrated production of a small molecule as well. So we can get into that as well. But the entire thing that we've been talking about is a lot of pharmaceutical companies right now are working on personalized medicine platforms. And the idea is that instead of scaling up, you have to scale out because, first of all, there are issues with the ways that their processes have in terms of scale. And then secondly, if it's personal, it's customized to each individual. So having small batch production of different therapeutics is what's really required. So that's number one. And an obvious application is, as Jeff has been mentioning, is military applications. There are many different ways that comes into play. But the thing that gets me most excited is pandemic response. So the U.S. government buys 300 million doses of smallpox vaccine and enough anthrax vaccine to handle 3 cities, and they just stockpile those, and they end up going to waste if you're lucky. And so imagine being able to make all of those on demand if and when they're required. So this whole pandemic response, and then what we could have is have, for example, freeze-dried cell-free systems that are sitting somewhere, and they could turn into any vaccine. So you have this capacity that you have that can really translate if you want to make, say, a vaccine against the coronavirus. As soon as you come up with a candidate that's good to go, you can scale immediately almost. So those are the things that get us super excited about this type of technology.
Great, fantastic, thank you. Now I have an interesting question for you, Jeff, because I'll preface it again, but I looked at your website. You have something that's called the Federal Machines Manufacturing Network. I would like you to explain a little bit about that, but to preface that, it's interesting. I think it's interesting if I interpret it properly, because that means there's a change in the supply chain. And in the management thinking of innovation, when there's different sources of change, such as a supply change, it stirs innovation. For example, a while ago, I saw a website that showed pharmaceutical thefts. It does happen. You get a big trailer car of certain types of medicine, and somebody hijacks the trailer and drives off. And this is mapped all over the United States. And the funny thing is, when you superimpose that with the major distribution centers across the United States, they actually overlap quite well. And what that is, that's the land-based distribution system of drugs distributing packages as they land from airports or ports that are ocean-based, and they distribute it around. But Jeff, you've got something that's called the Federal Machines Manufacturing Network, which may totally change that if it's run all over the place. And the supply then will be different, because they would be raw materials perhaps, and maybe hospitals may share, I don't know. Tell us about that.
Well, the concept for that is really an offshoot of the military again. So our military puts medical treatment facilities all over the world, in their bases, on their ships, and the like. So it's not unlike on a more bounded scale, kind of like in the United States. And so in the US, for example, the C-19 pandemic has shown us the vulnerability of our supply chain. So for example, New York City was running out of certain medications during their spike, just like Houston is right now, right? And those are things such as like are drugs that you need for ICU patients. The fentanyl we spoke of, midazolam we spoke of, the cisatracurium that is used, so on and so forth. The number of drugs that every ICU patient wants needs certainly when they're in the ICU, and they were running out of those things. That didn't mean the country was running out. It meant that New York was running out. And if you have a disrupted ability to deliver, let's say it was an earthquake, not a pandemic, there's going to be no drones flying because there's no electricity. You know, there's going to be no Amazon van pulling up because it's an earthquake. So the idea is, rather than trying to pre-position medicines that you may or may not need, such as I had in Afghanistan, it didn't mean I didn't have medicines. I didn't have the medicines I needed. So what you could do is pre-position our machines in areas that you think that are going to be vulnerable, and then those machines can be activated and make the medicines that are needed. So for example, you could have, in my mind, our machine is now the size of a refrigerator, but that's version 1.0. I mean, how big was your cell phone, Sam, when you and I were in high school, right? Aiden wasn't born yet, and Sari wasn't born yet, but you and I are old guys, and our cell phones were in bags. Remember that? We had a bag cell phone. That's not what it is today. So in my mind, I think that the machine will shrink down even more and be the size, seriously, of a home printer. you have something like that, you could pre-position that in fire stations, for example. That was my mindset. Pre-position a firehouse. Firehouses all have emergency medicine technicians there, EMT, paramedics. And should there be a crisis, it can make certain medicines that you would need. You don't have to make, antihypertensive. You don't have to make diabetic medicines, but you certainly, certain antibiotics you would need if there was a outbreak. There are certain, for example, antidotes for certain things if there was a terrorist attack, so on and so forth. So it's a very small number. And one of my machines could actually do that. And you could pre-position it, have it sit in the corner, and then, God forbid, a crisis happened and you fire it up. And the important thing is that resilience and flexibility I talked about. The resilience and flexibility is very important. You can't take out a distributed network like that. It's got too many heads to it. You blow up one firehouse, people go next door to another firehouse. Why do I say firehouses? Because you can walk to a firehouse. If you have a disaster, you don't want to be getting in anybody's car. You don't need to get in a helicopter. You want to walk to your supply, correct? And then the other thing about this is that we also have an opportunity now to create a system that is very flexible. So if you were in New York City and you were running out of fentanyl, you make fentanyl. If you were in Houston and you're running out of cisatracurium, you'd make cisatracurium, and the beat goes on. So you don't have to decide a priori up front what the shortage will be. It doesn't matter what it is. Whatever it may be, you go to the machine and you punch it, you turn it on. Now, as I said to you before, this is developed by the military. So subversion, falling in the hands of the enemy, we always worry about it. So it has built-in safeguards. I mean, if it's tampered with, it'll destroy itself, things like that. And so you have to build in, when you have a situation like this, you have to build in the appropriate safeguards and to prevent diversion, to prevent usurping the machine for nefarious reasons, so on and so forth. But because it's built for the military, we already think about things like that. We don't want it to fall into enemy hands. We don't want it to do other things, and so on and so forth. So those are all things you have to build in. These are issues that have to be addressed, but they're surmountable problems. They're engineering questions. They're not discovering new biology or new chemistry. I mean, Ada's discovering new biology. Sari's discovering new chemistry. I mean, these guys are brilliant guys. But I mean, here I'm just talking about making a machine and applying technology. So, I'm a bit of a hammer swinger, to be perfectly honest with you. And this is all hammer swinging.
So I have a question now where someone's actually this is very thought-provoking, is did we miss something? Because is there some other future of drug manufacturing technology or business model that we did not cover? So I will ask each of you whether there's something there, and maybe it's not. Maybe what you're doing, I said that your visions all started in 2016. Sorry, you became VP of R&D in 2016. Jeff, you became CEO of your company to take this vision forward in 2016. Aidan, your technology came out of the genesis of the work in 2016 of those people. So over time, these things take time to develop, but the acceleration means that it just exponentially gets faster as things move along, and you're driving each of those visions forward. Is there something else that might be out there that you're just, because you're not focused on it, that we should also be aware of, whether it's making things from plants or algae, or is it something about the model, which is about distribution in this world where globalization is changing, pandemics may be affecting things? Is there something else there? Let me go and start with Sari, and then Jeff and Aidan.
So I think that there is still a lot that can be done in this area. One simple example, I'll give you an example and I'll give you a framework to think about. One simple example is 3D printing of tablets, right? What Jeff just described, he has something which can produce the drug substance on demand. But once you have the drug substance in demand, you can just 3D print tablet. What Aidan is talking about is 3D printing of essentially molecules. You're chaining together the individual ingredients, the amino acids that make the protein. So 3D printing can happen at the molecular scale, 3D printing can happen at the atomic scale, it can also happen at the drug product scale. So I think that's something that needs to be thought about. That's very exciting. The second thing I'll say is all the innovations that Jeff and Aidan talked about so beautifully, it's very inspiring to hear all of these, are also very useful in R&D. Right? Because when we're producing dozens and dozens of compounds, dozens and dozens of candidate molecules, you need systems like these that can rapidly turn things around, and you can do very high throughput screening. But the very different take I've taken, my background is I've done work in semiconductor industry. I used to actually make display devices for cell phones. That's something I used to do in Silicon Valley. I did fuel cell technology, which is not very different from continuous manufacturing. Also, the US military is a big application for that. I've done a lot of work in automotive manufacturing, right? I think there is a lot of scope to bring the innovations that have really made those industries very efficient into pharmaceutical manufacturing. If the semiconductor industry can make millions of chips with absolutely low cost, miniaturize them, produce them with high degree of reliability, high degree of yield, I think there is a place for all of that to come into pharmaceutical manufacturing so that you can make drugs reliably at a high quality at a low cost. And I don't think the pharma industry is there yet. And so there's a long way to go in terms of making high quality medicines.
Thank you. Jeff, what else is there?
The things that Sari had talked about also open the possibility of truly personalized medicine as well. Sari is absolutely right. And right now we give a pill, same pill to everybody, and everybody's not the same. So this idea of personalized medicine really becomes possible with exactly what Sari said, that a person who needs a big pill, it may be in a different final formulation than somebody who needs a little bit of amount. So there's a real chance to do that. That's an obvious. Something that's not is we think about what are we trying to achieve here? I mean, when you give or take a medicine, what are you trying to do? You're trying to fix something that's not working, correct? I mean, that's fundamentally it. If you are taking medicine because something is disrupting your physiology, be it a C19 virus or a tumor or whatever. So in many of these situations, taking a pharmaceutical may not be the best way to fix this problem. In fact, a better way to fix the problem might be actually genetic engineering. If you had type 2 diabetes and you had a series of abnormal genes, wouldn't it be better just to fix the genes rather than take insulin every day? So I think one of the exciting areas to think about that would be very disruptive to what we're talking about is when, and I say the word when, they actually come up with the ability to actually make gene therapy safe and efficient. Right now, it's not safe and efficient. The big bugaboo here is you got to use a virus, an adenovirus, but you have to use a virus to deliver the therapy. And we all know what a virus can do if it's out of control. Just look out your window. So the issue here in my mind is the biggest breakthrough that could happen in terms of health is where we're not taking pills and we're not being vaccinated. Rather, there is gene therapy actually comes into its own, because we'll see a remarkable reduction in human suffering when that happens. We'll still have a place, by the way, for some therapeutics, painkillers, for example. We'll still have some place for some protein-based therapeutics. But the reality is if you want to know something that's going to disrupt the whole idea of even taking medicines, it's gene therapy.
Thank you, Jeff. And over to you, Aiden. We're running out of time, so let us know what we're missing, what's in the future. And
I'll make this really quick. I think one thing that can make all this a reality are regulatory innovations, because we really need to innovate on part of how we're going to get these into the market legally. So that's the whole thing. Like if you have these small boxes, CGMP approval, as far as I know, at least in Canada, is for a building. It's not for a box. So, how do we go around that? I think new expression chassis are on the horizon. We've been far too long stuck with E. coli and CHO cells for manufacturing most of our products. Also, companies like us will start acting as utility companies to take part of the hard work of fermentation out and do it upstream of things, so that people can basically just run the processes with those systems. And then lastly, cell therapy. So currently, 95% of the cost of growing those cells is the media, and 99% cost of that is the cytokines and growth factors that go into it. And these are ingredients that go into cultured meat industries. And when those scale up, the cost of those will drop dramatically. So cell therapies will become a lot more accessible to the general public.
And with that, that's a great thought. Maybe we will do our next Future of the Planet series on something like that. Thank you very much, Aiden, Jeff. Sari, I wish you all the best with your visions because we are all here to save lives. To everyone that's watching, may this be an inspiration for what you do. Thank you from all of us.
Thank you so much.
Thank you.
Nice day, everyone. Thank you.
Take care.